day, everyone. I am Camila Simbahan, and I currently took Master of Science, major in horticulture, and minor in soil science. Our group would like to discuss the assigned type of fungi given to us, which is the Basidium mycota. I am going to start with the introduction, then I'll discuss the definition of Basidium mycota and its beneficial Basidium mycota we've chosen, which is the Lipilum shimiji, and I'm also going to discuss the important rules of Lipilum shimiji with Miss Corey, while Mr. Oliver and Miss Bernal will discuss about the pathogenic Basidium mycota, Emilia bastratrix, treatments of coffee leaf rust, and state of the conclusion and acknowledgement. So Basidium mycota are known as large and diverse phylum of fungi or kingdom fungi. It is considered as the monophyletic sister group to the Skomycota. It- Basidiomycota are typically filamentous fungi composed of hyphae. Most species reproduce sexually with a club-shaped spore-bearing organ, basidium, that usually produces four sexual spores, or basidiospores. So the most diagnostic feature of basidiomycetes is the basidium or basidia, a generally club-shaped cell where nuclear fusion or karyogamy takes place and the structure upon which the sexual spores or basidiospores are produced. Basidia are born on complex multicellular fruiting bodies, which are large and conspicuous in all but the yeast, rust, and smuts. So here we are at the beneficial Basidium mycota. Like phylum Shimiji belong to kingdom fungi, phylum Basidium mycota, class Agaricomycetes, order Agaricales, family Lipolaceae, genus Lipulum, species Shimiji. So Lipulum Shimiji is commonly known as Han Shimiji. Shimiji is shaped like mushroom. It has an umbrella and a handle. L. shimiji is an ectomycorrhizal fungus that is widely distributed in East Asia and also present in the northern regions of Europe. This fungus grows in pine forests, often near man-made roads, cultivated experimentally in pure culture using selected strains capable of growing subreptically. Traditionally, fruiting bodies of L. shimiji have been collected from the wild, but fruiting of L. shimiji is now relatively uncommon and cannot keep up with the increasing consumer demand. As a result, methods of cultivation are being developed for commercial production in Japan and in other countries. In this work, techniques were developed to cultivate L. shimiji and coniferous seedlings using a pure culture inoculum. So, so L. shimiji is a culinary delicacy in Japan. It is considered amongst all edible mushrooms to have the best taste and to be second only to Tricoloma matsutake and rice. So it is a culinary delicacy in Japan. Shimiji mushrooms are considered edible and have a mild flavor and aroma that enhances when cooked. They can be used in soups, simmered dishes, deep fried foods, and seasoned rice. We have also other recipes of El Shimiji, which is the shrimp and mushrooms with rice, curry seasoned cauliflower and mushrooms, salmon and shimiji mushroom cream stew. So El Shimiji, as an ectomycorrhizal fungus, helps plant absorb nutrients from the soil in exchange for food to assimilate. This type of symbiosis is mutualistic and is one of the most common types of symbiosis between fungi and plants. El Shimiji is known to be an ectomycorrhizal fungus associated with plants in the Fabaceae and Pinaceae families, and mycorrhizal symbiosis is one of the most abundant types of symbiosis between fungi and plants. This type of symbiosis is basically mutualistic in that fungi help plants take up soil-derived nutrients such as phosphate and nitrate in exchange for food to assimilate.
I will be discussing about the health benefits of Leiophyllum shimiji or the shimiji mushroom. So the mushroom provides protein, it has dietary fiber that aids in digestion and maintains gut health. It also has B vitamins such as vitamin B2, B3, and B5. It also contains vitamin D and contains minerals such as potassium, copper, and phosphorus. So it also has antioxidants such as ergothioneine, which helps neutralize free radicals in the body. So ergothioneine is an amino acid that is mainly found in mushrooms and red and black beans. So this amino acid can reduce oxidative stress and may help prevent chronic diseases, including certain cancers and cardiovascular issues. So another health benefit of the shimiji mushroom is it contains polysaccharides, particularly beta-glucans, which have been shown to enhance the immune system. So beta-glucans enhance the immune system. These compounds, this beta-glucan, stimulate the activity of immune cells like macrophages and natural killer cells, improving the ability to fight off infections and potentially reducing the risk of tumors. And another health benefit of the shimichi mushroom is it has anti-inflammatory properties, which as you can see in this picture of a research, this research shows the anti-inflammatory properties of certain edible mushrooms, such as which includes Leiophyllum shimiji. About cultivation of the mushroom, since in Japan this mushroom is a delicacy and it has a very high demand, but this demand cannot be met anymore because this mushroom, which grows on boreal forests, has, since there has been, due to deforestation, they have now less places to grow. They had their habitat is getting little. So in Japan, they have been researching on how to cultivate the mushroom artificially. So there have this one here, this production of fruit bodies of a mycorrhizal fungus in pure culture is one study that has successfully obtained the fruit body through artificial cultivation using barley grain without the host plant. So it was done by Akira Ota. That's all. Thank you.
Now, let us talk about pathogenic Basidium mycota, or a fungi that causes plant diseases. So, an example is a rust fungi. This is an obligate parasite that needs a host for them to survive and complete their life cycle. So, among the plant pathogenic fungus out there, in terms of species, rust fungi is a great category. So, the rust-related diseases, these are a major worry for our global food security, since they impact many agriculturally important plants, including coffee, corn, as well as they could infect some of our fruit trees. So Humilia vastatrix is a causal organism of a coffee leaf rust. So it comes from the kingdom fungi, phylum Basidiomycota, class Posiniomycetes, order Posinialis, family Zaguaniaceae, genus Humilia, and species Vastratrix. And identified as the cause of a coffee leaf rust in Ceylon and Eastern Africa in 1869. So when it reached Philippines in the 1880s, there was a great coffee industry setback because there was a decline or a decrease in coffee yield production during that time in the late 1891. Cellular fungi that causes the coffee leaf rust disease. So its symptom is the chlorotic spot or the yellow-colored spot under the coffee leaf, as well as the iridonosporic sori in the lower leaf surface. It causes the leaves to drop and subsequently causes the killing of the coffee plant because there is a severe defoliation in plants. So it is a hemicyclic fungi with iridonosporic life cycle. It has uridinospore as its spore produced, and with the help of hyphae, it ruptures the plant cuticle.
How does rust fungus Hemilia vastatrix infects plants? So this rust fungus infects plants by releasing iridonospores, which is a fungal spores responsible for the infection, and it is dispersed by wind, rain, or human and then land on the leaves of the host plant. So where in fact, some studies revealed that coffee leaf rust caused by Hemilia vastatrix is spread by rain's flash from the infected leaf litter. So their findings suggest that rain can still transmit spores from the leaf litter into the uninfected leaves, and it will significantly increase its infection. The figure illustrates the disease cycle of Hemilia vastatrix. So its infection process starts with the initial phase, which is the spore landing and germination. Uridonospore, the fungal spore responsible for the infection, will land on the lower surface of the coffee leaf. So they appear a small rounded structure under the scanning microscope, which can be observed in the upper center of the figure. The uridonospore germinates within about 17 hours after the inoculation. It will produce a germ tube which extends across the leaf surface in order to locate the stomata, a tiny openings of the leaf, and it will form a specialized structure called an aprisorium. So this aprisorium is important or essential for penetrating the leaf. The second phase of the infection process of Hemilia vastatrix is the stomatal penetration. So at around 24 hours after inoculation, the aprisorium positions itself over the stomata. So from there, a penetration of hypha, a thin fungal filament, enters the plant through the stomatal opening. The penetration of hypha grows between the cells within the plant tissues, and it will develop hostoria, which are specialized structure used to extract nutrients from the plant cells without killing them. So this can be observed in the third and fourth figure in the right side. After penetration, the fungus spreads extensively inside the leaf tissue. Intercellular hyphae extend throughout the epidermal and mesophyll layers, while hostoria invade individual plant cells to absorb nutrients. So as shown in the figure, in the center below, the fungus colonizes spongy parenchyma cells deep within the leaf. So it is visible under the scanning electron microscope as extensive intercellular hyphae and hostoria. So the last phase of the infection process of Hemilia vastatrix is the spore formation and release. So around 21 days after inoculation, the fungus produces new uridonospore within specialized structure called sori. So these protrude through the stomata in a characteristic bouquet shape, as seen in the upper left of the figure. The sori releases uridonospore into the environment, and it is visible under scanning electron microscope, and it is the sign which signifies the complete cycle and allows the fungus to spread to the new plants. So this cycle is important because it explains how Hemilia vastatrix invades the coffee plant and extract nutrients from it. So it is important to understand these stages in order to design a control strategies and preventive measures in targeting the spore before the germination and enhancing leaf resistance to the fungal penetration. There are preventive and control measures of coffee rust caused by Hemilia vastatrix. First is good sanitation practices. Keeping the coffee farm clean helps prevent spread of the fungus. For example, regularly removing fallen leaves and infected parts of the plant can reduce areas where the fungus might thrive. Second is to maintain tree health. So in what way? By simply removing weeds. Because removing weeds ensures the coffee plants gets enough nutrients and are not stressed by competition from other plants. Healthy coffee plants are less likely to get sick, so that's why it's important to maintain its health. Next is to pruning coffee plant in order to reduce humidity and increase airflow. So cutting back some branches allows more air to flow and reduce moisture around the plant. This creates an environment less favorable for the fungus which thrives in damp condition. Next is remove old, weak, and infected crops. Plants that are weak or already infected can spread the fungus to healthy plants. So removing them ensures that the disease does not spread further. So lastly, prior to application of any products, it is important to consult local advices. So not all fungicides or treatments work the same way in every region. So it is best to seek advice from local experts who understands the specific condition of the farm before applying any products. So some studies suggest to use a resistant varieties and biopesticides in controlling the rust fungus caused by Humilia vastatrix. So use of resistant varieties is a long-term and sustainable solution. So these varieties have genetic traits that make them less susceptible to infection. Just for instance, the coffee Liberica, which is native in Western and Central Africa. So it is now cultivated locally in the Philippines and Malaysia. So it is resistant to coffee rust disease, and this work in the way of inhibiting the fungus ability to penetrate the leaf or reduce its capacity to grow and spread within the plant tissues. So what are the benefits of using resistant varieties? So first, it can reduce dependence on chemical controls. It also provides a more consistent yield under high rust pressure. And lastly, and most importantly, it requires less maintenance compared to chemical and cultural controls. Another preventive measures or control measures that some researchers suggest is the use of biopesticides. Biopesticides are natural or biological agents used to manage pests and diseases. So for coffee rust caused by Humilia vastatrix, biopesticides offer an eco-friendly alternative to chemical fungicides. So just for instance, the use of essential oils and antagonistic microorganisms as eco-friendly alternatives for coffee leaf rust control. So these products often contain beneficial microorganisms like fungi or bacteria, plant extracts, or naturally derived compounds. So how they work? So biopesticide works by suppressing the fungus or disrupting its growth. So it reduces the spore germination or it prevents its ability to penetrate coffee leaves. So some benefits includes it is safe for humans, animals, and environment. It also helps in maintaining soil and ecosystem health. And lastly, it reduces the risk of developing fungicide resistance. There are biological and non-biological treatments of coffee leaf rust caused by Humilia vastatrix. Some biological includes the application of chitosan to coffee plants, which is a natural and biocompatible and biodegradable polysaccharides derived from the exoskeleton of crustaceans such as shrimps and crabs. Another is the use of microparasites such as Digitopodium, Himalae, Digitopodium canae, and others. So another microparasites that could be used to target Himalaya vastatrix is the Lecanicilium lecanae. On the other hand, non-biological treatments of coffee leaf rust caused by Humilia vastatrix have also been explored. It includes selective breeding of resistant coffee plants, and along with it, protectant fungicides such as copper hydroxide are commonly used as preventive measures against the pathogen. Treatments involving salicylic acid have also been explored after being validated by their success against other plant pathogen. This treatment was discovered to be of use in reducing both severity and incidence of coffee leaf rust. In fact, it holds the potential to be more effective than the traditional treatment options of copper hydroxide and other fungicide. However, we should always bear in mind that no matter how many cure or treatments presented in that particular diseases or pathogen, prevention is better than cure. That's the least we can do. And I do believe that the more you can prevent a particular diseases, the better.